Was there any chance of phone here?
Not that I noticed.
All right. Well, thanks very much.
Yeah. Okay, all right. Well, good afternoon, everyone. So, a couple housekeeping things before we continue on with our lecture here. Perhaps most importantly, so we have our fifth and final term quiz coming up next Monday, Tuesday. So don't forget about that. Just before I got here to the lecture this afternoon, I posted the practice quiz questions from last week's content. I also posted an additional set of questions from content covered earlier in the course. Because of reading week, we only have covered 1 week's worth of content that would be eligible for the term quiz next week. So we're going to try and mix in a few questions from content earlier in the course, mostly immunology, maybe 1 or 2, 3 at most kind of questions from there. Those practice quiz questions that I posted that are called additional questions, they're all immunology focused. So just have a look through there, and that might help to remind you about some topics from earlier in the course. Probably good for us to get back to thinking about some of that earlier course content anyways, because our final exam is not too far away. In reality, we're less than a month to the final exam, which is on December the 8th. So our final lecture is the 28th of November, that Friday, and then, a little over a week and a bit, and then we're into the final exam. So again, probably not a bad idea for us to start thinking about some of that older course content, bringing it back to the forefront of our minds to get ourselves prepared for that final exam. The deadline for finishing up Short Communication 1, at least if you're interested in those 2% bonus marks, is on Friday. Again, the TAs are working hard to mark things. If you have had your submission sitting around for more than 3 business days, feel free to let me know about it. I do realize that if you've emailed me in the last couple of days, I have not yet responded. I have not had a chance to get to my emails. A large part of that was being here all day on Sunday for the fall campus day, and then the drive home that day was a miserable disaster. There was a transport truck that had lost control, was on fire. The highway was shut down. Me and thousands of other people, it seemed, were on this snow-covered dirt road, snaking our way around the 401 accidents, and so couldn't do a lot of email answering because of that. But I am working on it. I will get to those emails as quickly as I can if you've sent me anything. Okay, those are all the things I wanted to say. Questions about anything before we jump into the lecture here? Everybody say okay? Okay. All right. So we ended the lecture on Friday with this slide talking about glutamate receptors. And just to kind of remind you of some of the things that we had discussed there. So there are 2 glutamate receptors, 2 very common ones in the central nervous system. One is the AMPA receptor, which is named for this agonist of the receptor called AMPA. And the other is the NMDA receptor, so named for its agonist as well, which can activate it, NMDA. And what we saw is that, and as it says at the top right there, these receptors often coexist. And so let me just kind of walk you through again kind of how glutamate, what does it do when it binds to these receptors? What's the response we're going to get from the postsynaptic cell? So here's our presynaptic cell releasing glutamate. Here's that glutamate binding to the AMPA receptor on the postsynaptic cell. The AMPA receptor is itself a sodium channel, a gated sodium channel. So when glutamate binds to the AMPA receptor. That will cause the gate on that channel to open, and that's going to allow for sodium to come into the cell. And so, of course, we know that as positively charged sodium comes into the cell, that will make the membrane potential more positive. So this will be a depolarization, an excitation, if you will. So that's the effect that glutamate is having via the AMPA receptor. Through this other receptor, we said that there are kind of two things that are required for it to become active. First is that, yes, glutamate has to bind to the NMDA receptor. That's one of the two things required to activate this receptor. But we also noted that just glutamate binding alone is insufficient because you have this magnesium ion that is actually blocking the NMDA receptor. And we said the reason that it's there blocking is because the equilibrium potential for magnesium is negative 40 millivolts. The resting potential is typically somewhere around negative 70 for a neuron. So under resting conditions, magnesium is not at equilibrium. Magnesium wants to be coming inside of the cell here to try and bring it closer to its negative 40 millivolt equilibrium potential. So here we have a magnesium ion trying to enter this postsynaptic cell, trying to come in through this NMDA receptor, but it can't fit. So all it ends up doing is blocking that receptor. So once glutamate binds to the NMDA receptor, that's one thing required to activate it, but we also have to coax that magnesium ion out of there, and that's typically done through the depolarization caused by the AMPA receptor. Because if you have that sodium coming in through the AMPA receptor to depolarize that postsynaptic membrane, once that membrane potential gets above, more positive than negative 40, then the magnesium is going to want to move in the opposite direction. It's going to want to try and leave the cell. It's going to want to leave that NMDA receptor. And then once it does, it unblocks the receptor. The receptor is active because glutamate's bound. And now calcium is free to actually move through that NMDA receptor. And so that's what happens here once you have this fully activated, unblocked NMDA receptor. Calcium passes through it and enters into the postsynaptic cell. And then as it says here, calcium will typically then act as some kind of second messenger, right? It'll activate some kind of pathway within that postsynaptic neuron, and that will be the nature of the response that you'll get from that postsynaptic neuron. So glutamate in this context is thought of largely as being excitatory, right? It's causing depolarizations. It's allowing calcium to come in and activate some kind of second messenger system. In the central nervous system, and we said this last week as well, we typically think of glutamate as being an excitatory type of neurotransmitter within the central nervous system. Okay, so what I want to talk about next here, kind of moving on from where we were last week, is to talk about this concept of excitotoxicity, which is basically the idea that if there is too much glutamate signaling happening to a neuron within the nervous system, that glutamate stimulation can actually overexcite the neuron to the point where the neuron actually dies as a consequence of that excitation. So that's what this term excitotoxicity means, right? Basically, excitation as a toxic agent, an agent that can lead to neuronal death. So what causes this? Well, it's largely caused by the calcium influx that occurs in response to glutamate signaling, as we just saw, right? So glutamate is going to activate the NMDA receptor and allow calcium to come in, as they're depicting here, calcium flooding into this postsynaptic neuron. And so, again, if we have a lot of glutamate and we have a lot of activation of the NMDA receptor, then we have a lot of calcium potentially coming into this postsynaptic neuron. Yes, calcium can serve as a second messenger. It can activate signaling pathways within the neuron, as we just discussed. But as we had also learned earlier in the course, and this came up as we were talking about multiple sclerosis, we know that prolonged elevated levels of calcium can actually cause the death of a neuron. Really, it can cause the death of any cell, but in this context, we're thinking about neurons. And that's because, as they depict here in this figure, calcium does things like turning on proteases that will then degrade the proteins that are within the cell. Calcium is an activator of phospholipases, which will then degrade the phospholipids that make up the cell membrane. And calcium is also an activator of nitric oxide synthase, which, as we saw earlier in the course, produces nitric oxide, which can react with other kinds of reactive oxygen species to give you these really reactive peroxynitrate molecules that can then actually cause significant oxidative damage to the biological molecules that make up the cell. So that's what they're kind of talking about when they say here, increase free radicals. So this is the nature of excitotoxicity. If you've got a lot of glutamate signaling acting on a postsynaptic neuron, and you've got a lot of these NMDA receptors open as a result, allowing calcium to come in and get to these higher levels for long periods of time, the effect that calcium can have on that postsynaptic neuron is ultimately to, again, upregulate proteases, phospholipases, facilitate peroxynitrate production, all of which is going to ultimately culminate in the death of the cell, right? So that's what we mean by excitotoxicity. Let me give you a specific example of a disorder where excitotoxicity is kind of at the forefront. And the example I'll give you here is ALS. Right? ALS stands for amyotrophic lateral sclerosis. This is basically a disorder where motor neurons die, again, often because of excitotoxicity. And as the motor neurons die, they, of course, become unable to stimulate their corresponding muscles. And so we start to see muscles shutting down, right? Not being able to be stimulated, undergoing atrophy and deteriorating. And this can culminate in things like paralysis, so an inability to move. It can ultimately culminate in respiratory paralysis, an inability to breathe, and so a fairly catastrophic and fatal disorder. You're probably familiar, or maybe you're too young to still be familiar with this, I don't know anymore, but the Ice Bucket Challenge, that was a big thing for a period of time, of course, was an ALS fundraiser, so that's one of the things that people often think about when they think about ALS. ALS is also known as Lou Gehrig's disease, and most of you are way too young to know who Lou Gehrig was. He was an iconic baseball player with the New York Yankees back in like the 1930s and 40s. His career was cut short by ALS. He was at the height of his career. You can see videos of him on YouTube standing in front of the crowd at Yankee Stadium, basically announcing his retirement from baseball because of Lou Gehrig's disease, ALS, and ultimately he died not long afterwards. If you're not familiar with Lou Gehrig, maybe you're more familiar with Stephen Hawking, also, of course, famously had ALS. He had a version of ALS that was a lot more slow in terms of its progress, so he lived a much longer life than Lou Gehrig did. But if you know anything about Stephen Hawking, it gives you at least some way to visualize the impacts that ALS can have on a person's body. In any case, I want to think about how exactly can excitotoxicity occur in the context of ALS, leading to this motor neuron death and ultimately to these paralyses of the body. I should start by saying that there are many different underlying causes of ALS, so we're not going to obviously explore this in tremendous depth. But the one that I'll focus on here is something called SOD1-linked ALS. So it's called this because it has been linked to a mutant version of SOD1. So SOD stands for superoxide dismutase. So it's an enzyme that is meant to take superoxide, this free radical that's produced largely by your mitochondria, and it's meant to convert it over into hydrogen peroxide, and then catalase can kind of come in there and break that peroxide down into just oxygen and water and protect you, therefore, from the damaging effects of this compound. What happens in folks who have this mutant version of SOD1 is that it actually starts to kind of wind up in the wrong places within your cells and can actually increase, rather than helping to decrease ROS and free radical production, the mutated version gets kind of localized within the cell and actually facilitates more aggressive production of free radicals and reactive oxygen species. And this becomes an especial problem in the astrocytes because, as we learned last week, astrocytes have these glutamate reuptake transporters that they can use to remove glutamate from the synaptic cleft and thereby terminate its excitation on the postsynaptic neuron. What happens when you have this mutant version of SOD1 that's basically facilitating more free radical or ROS production is that the glutamate transporters, these reuptake transporters, they are particularly vulnerable to free radical attack. They have a lot of cysteine in their structure to kind of hold the protein in its correct three-dimensional shape. And so when you have all these free radicals creating this highly oxidizing environment, this can actually disrupt the normal bonding that these different cysteines do with each other. And ultimately, this means that you have all these free radicals that are basically altering the structure and therefore the function of these glutamate reuptake transporters in a way, as they show here, that prevents them from being able to take up glutamate from the synaptic cleft. So you have your motor neurons here releasing normal amounts of glutamate, right, when they get stimulated, but because of this increased reactive oxygen species free radical production in these SOD1 mutant astrocytes, that ROS damages the glutamate reuptake transporters to the point where glutamate cannot be as effectively removed from the synaptic cleft, so its concentration gets a lot higher. It stimulates those NMDA receptors to a much greater degree, allowing for far more calcium to come into the cell, and ultimately that postsynaptic cell will die based on the mechanisms that we talked about just moments ago. So we can see here an example of a specific disorder in ALS where we've got motor neurons dying, right, because they're being overexcited by glutamate, because glutamate is not being effectively removed from the synaptic cleft because of damage to these glutamate reuptake transporters that's being caused by this mutant SOD1 that's actually facilitating more ROS that's causing damage to those reuptake transporters. So again, too much glutamate signaling to a neuron can ultimately lead to its death, as we've now noted. I want to give one example here of a glutamate receptor that's not excitatory. Last week we said that most often glutamate is thought of as an excitatory neurotransmitter, but there are examples of where glutamate can actually be inhibitory. And so I want to give you an example here of a glutamate receptor that is inhibitory in nature. The glutamate receptor I'm going to introduce you to here is called the mGluR6. Okay, so this is our third glutamate transporter that we're learning about now. What you can see here is that this mGluR6 is clearly a metabotropic type receptor because you can see that here's glutamate binding to that receptor, but then here, way over here, are the ion channels whose status, shall we call it, is impacted by the binding of glutamate to that mGluR6 receptor. Okay, so this is a metabotropic type of receptor where your receptor and the ion channels that it affects are separate entities within the membrane. So what happens here is glutamate binds to the mGluR6 receptor. This activates an associated G protein. So there's the G protein kind of being activated and splitting apart. And then this G alpha part of the G protein, you can see it goes over here, and it closes this thing called the transient receptor potential channel, or the TRP channel. And you can see here this is depicted as a sodium and calcium permeable channel. So if we close that channel, we're going to block the ability of both sodium and calcium to come into the postsynaptic cell here. And so as fewer of these positive charges come into the cell, that's going to lead to the hyperpolarization of that cell. And so now you can appreciate why this is an inhibitory glutamate receptor, because it's causing hyperpolarization, right? It's decreasing the likelihood that this postsynaptic cell is going to undergo any kind of action potential of its own. Let me give you a sense of where this mGluR6 receptor plays a role in our bodies. It actually has an important role to play in terms of vision, the detection of light in our environment. So what you have here at the top, it says you have the presynaptic terminals of rods. So you're probably familiar with the rods and cones that are in the retina of your eye, right? These are the light-sensitive neurons within the retina of our eye that have a role to play in terms of our vision. And so what it's showing us here is when it's dark, so when a rod is not detecting any light, right? When it's dark, the rod cell, the rod neuron, is actually releasing glutamate under those conditions. Kind of a little weird to think about. Usually we would think about it the other way. You'd think, oh, if there was light, we'd release a neurotransmitter, but it's actually the opposite. These rod neurons, these rod cells, when they do not detect any light, when it's dark, they will release glutamate. And then down here, you have these other cell types called on-bipolar cells. So they're part of the series of neurons that's going to kind of relay information back to the visual cortex of the brain so that you can perceive the light that you're detecting and have vision. And what you'll notice here is they mention that there's this metabotropic glutamate receptor. This is our mGluR6 receptor that's expressed in the dendrites of those on-bipolar cells. And so in the dark, when our rods are releasing glutamate, that glutamate is binding to this mGluR6 receptor. And in this case, it shows us that it's activating this G protein here. This G protein, in turn, is activating phosphodiesterase. So this is an enzyme that breaks down cyclic nucleotides. So here they're drawing your attention to cyclic GMP, but we've also seen cyclic AMP before, so different cyclic nucleotides. But phosphodiesterases, when they're active, they chew those cyclic nucleotides apart. So here you can see the level of cyclic GMP goes down, and that's significant because cyclic GMP is one of the activators of this TRP channel here, right? So what they've called here a sodium channel is this TRP channel, and in the absence of cyclic GMP, this channel is now closing, right? And so this on-bipolar cell, because the sodium channel is closing, will then hyperpolarize. And so it will not send an action potential back to the visual cortex of the brain. And so the absence of that action potential coming to the visual cortex tells us that that rod is currently experiencing no light, darkness. And that's what we would see then. We would see dark in that area of the field of view where that rod was located. But then, when that same rod is exposed to light, it actually stops releasing glutamate. So there's no binding then to that mGluR6 receptor. That G protein is no longer active, so the phosphodiesterase is no longer active. Cyclic GMP levels build up because they aren't being degraded, and then that activates that TRP channel, allowing sodium and calcium to come in, causing depolarization and therefore excitation of that on-bipolar cell, and that then will lead it to generate an action potential, and then that action potential is ultimately propagated to the visual cortex, and now we're going to see light in that particular part of our vision that is associated with that rod. So yeah, we can see in this context here, a little bit of a strange, unconventional system, but we can see the role that this mGluR6, this inhibitory glutamate receptor, is playing here in terms of our vision, right? That when it's dark, the glutamate that is released by our rod cells is ultimately causing inhibition of those on bipolar cells, ultimately leading our visual cortex to perceive that darkness due to a lack of stimulation coming from that on bipolar nerve. Okay, let's move on to our next amino acid neurotransmitter that I want to talk about here, which is GABA. We talked about GABA a little bit last week. We talked about the GABA B receptors in the context of presynaptic inhibition. We're going to talk today about GABA A receptors, which are quite a bit different. So GABA A receptors are ionotropic receptors, meaning that they are themselves ion channels. And so what you can see here is the particular ion that they are permeable to is chloride. So the GABA A receptor is itself a chloride channel. And as they're trying to show here, when GABA binds to the GABA A receptor, that will cause the gate on this chloride channel to open, and that will allow chloride to come into the cell. Now, we talked about chloride way back. I mean, either it's going to cause hyperpolarization of the cell, right, as this negatively charged chloride comes in, or often, as we saw earlier in the course, it might just stabilize the membrane potential kind of at its resting level. Regardless of which of those two things it actually does, overall, it's making it harder now to excite this neuron, so this is an inhibitory effect. And we often think of the GABA A receptor and the GABA neurotransmitter itself as being inhibitory in nature. Okay? Again, there are some exceptions to that, but generally speaking, 99% of the time, GABA and the GABA A receptor is going to have this inhibitory effect on the postsynaptic neuron. Now, what's beautiful about this figure here is it shows us that GABA is actually not the only compound that can bind to the GABA A receptor. There are actually lots of clinically, pharmacologically relevant compounds that can bind to this same receptor, and so I want to talk about these. I'm just going to go kind of around the horn here for the most part. So we'll start down here with ethanol. And ethanol is described as an agonist of the GABA A receptor, which means that it binds to it and has the same, has that same activating effect on the receptor that GABA has. So ethanol, I mean, you've probably learned about it since you were, in elementary school, talking about ethanol as a nervous system depressant, right? You know that if you, get yourself drunk, you're going to have a hard time, walking around. Your judgment's not going to be so great. That's because you are effectively reducing central nervous system activation, right? Because you have this ethanol that's an agonist of this inhibitory GABA receptor. So we can explain much of the kind of neurological effects of ethanol in terms, again, of reducing our judgment, making it so that it's harder for us to walk in a coordinated way, all these things. We can understand that because we can say, hey, ethanol is a GABA A receptor agonist. It's basically working to inhibit central nervous system signaling, and this is why our central nervous system becomes less responsive to stimuli, why we can't process information as well, why we can't coordinate our balance, and all these things. Next up here are the barbiturates, which are also described as agonists. So barbiturates, I give an example here, like phenobarbital. These, again, because they're agonists of the GABA A receptor, they should have that sort of same effect of ethanol. They're going to reduce central nervous system activity. And so what barbiturates were historically used for, because they're not really used in human clinical practice as much now as they once were, but if they're an agonist of the GABA A receptor, so they're going to cause inhibition of central nervous system activity, this helps us to explain what they used to be used for very commonly, which is things like reducing anxiety, right? So if you think of anxiety as being kind of the consequence of an overexcited nervous system, then being able to inhibit central nervous system activity via barbiturates could reduce anxiety. They were very often used as like anti-seizure or anti-epileptic medications. Again, if we just perceive seizures as the consequence of overexcitation of the nervous system, we can understand how a GABA-A agonist would be useful to treat that. And they were also used to treat things like insomnia, right? The inability to fall asleep. Because again, part of what allows us to go to sleep is our nervous system, going into this kind of, quiet, inactive state. And so if your nervous system your central nervous system in particular is too excited, right, then it's going to be hard to sleep. So you could use barbiturates as a way to quiet and inhibit the central nervous system to promote sleepfulness. Ultimately, barbiturates are hardly used for these purposes anymore because they're very highly addictive, and really the safety margin between their kind of clinically effective dose and their fatal dose is a little bit too narrow for comfort. So by and large, they've been replaced now with a different class of GABA-A agonists called the benzodiazepines. I give the example there of Valium, which is one that people are often familiar with. So these are used for the exact same purposes, right? Because it's an agonist of the GABA-A receptor. So you can use them for reducing anxiety, treating insomnia, anti-epileptic, and all these things. But they're not nearly as addictive, and they're typically a little bit safer to use. One of the things I'll kind of point out here, and I'll point it out right now, is just a note that, look at the barbiturates and where they bind to the GABA-A receptor. It's not at the same spot where GABA binds, it's at a different site. And then notice that the benzodiazepines bind somewhere else entirely. So all these different GABA agonists that we are talking about here that interact with this GABA-A receptor, they don't even all bind at the same location. This GABA-A receptor is kind of cool, where it has all these different binding sites within it that we have exploited with these different classes of drugs. But in terms of thinking about, like barbiturates and benzodiazepines, they have a lot of the same properties. I mean, they're both GABA-A agonists. We use them, again, to kind of quiet the central nervous system, to treat anxiety and insomnia and so forth. But the reason that they can have similar effects without having exactly the same, pharmacological safety profile, for example, is partly because they are binding to different locations on the receptor, right? We have the same overall effect, but they're binding through different locations, which likely accounts for their different properties. The other thing I wanted to mention here, going back to barbiturates for just a moment, is one of the places today where barbiturates are still used is for kind of euthanasia, lethal injections, because if you overdose someone with barbiturates, be it phenobarbital, be it something like thiopental, if you overdose someone on these drugs, obviously it's going to basically shut down nervous system activity and basically, kill the individual by shutting down their nervous system. So in the context of, like I said, lethal injections, euthanasia, and I'm not talking just euthanasia of humans in the context of, say, medical assistance in dying, but also euthanasia of even just animals. If you're putting down the vet is putting down an animal or something, thiopental and other barbiturates are still routinely used for that purpose. At the very top here, it talks about neurosteroids, and I want to talk specifically about these two neurosteroids here, DHEA and DHEAS. These compounds are both precursors, DHEA, I should say, is the precursor to both testosterone and estrogen synthesis. And what's interesting here is it says that DHEA is an agonist of the GABA-A receptor, and then DHEAS, the S here standing for sulfate. So it's a sulfated version of DHEA. The DHEAS is actually an antagonist of the GABA-A receptor, meaning it binds to it and stops it from being able to activate, right? Blocks this chloride channel from activating. So what's the significance of this? Well, one significance that I'll bring up here is there is this hypothesis called the GABAergic deficit hypothesis of depression. So we talked a little bit last week about serotonin, lack of serotonin signaling in the brain as one causative, underlying cause of depression. Another underlying cause of depression is a lack of GABAergic signaling, not enough GABA binding to GABA receptors. And so how is this relevant specifically to DHEA and DHEAS? Well, I'm going to make it relevant by talking about postpartum depression. So we're talking about this very specific type of depression experienced by mothers sort of during their pregnancy and after their pregnancy, which has important consequences because mothers who experience postpartum depression, oftentimes they kind of neglect and fail to bond properly with their children. Obviously, that can be difficult for them. It's very difficult on the child. So, yeah, this is a very important subcategory, shall we say, of depression worthy of spending a little time thinking about. The GABAergic deficit hypothesis is often put forward as a possible explanation for postpartum depression. And here's what I mean by that. When a woman gets pregnant, the fetus will actually produce a lot of DHEAS. Okay? Mostly comes from the fetal adrenal gland. It'll pump out this DHEAS. And what happens to that DHEAS is it goes to the placenta, and then the placenta is supposed to pull the sulfur group off. The sulfate, that S, is supposed to be pulled off, so that what goes into mom is DHEA, and then mom is able to use that DHEA to produce estrogen. And that's necessary because estrogen is such a critical hormone in pregnancy, right? It basically helps to adapt the body to the pregnancy that is occurring. You know, if I just give one example, it sort of like, for example, softens up, the pelvic ligaments in a way that'll make it easier for the fetus to be delivered once it reaches term. So we've got this DHEAS that is being produced in abundance by the fetus itself and its adrenal glands, being desulfated by the placenta so that DHEA will go into the mom and be turned into estrogen to support the pregnancy. Well, there is evidence that in women who experience postpartum depression, there's evidence that their placenta fails to take that sulfate group off properly, fails to get it off of there. And so what ends up happening is DHEAS makes its way in abundance into mom's body, and some of that gets to the central nervous system and antagonizes, right, inhibits these GABA-A receptors, leading to a GABAergic deficit and thereby the onset of depression. So that's one hypothesis anyways around postpartum depression, but it also kind of nicely links to our GABA-A receptor that we're thinking about here. Lastly, another antagonist I'll bring up is picrotoxin. This is actually a toxin that is derived from the seeds of this plant here called Anomitra cocculus. So the seeds are rich with picrotoxin, which is an antagonist of the GABA-A receptors. You can see how it works. It effectively actually binds to a location on the interior of the channel here, thereby making it difficult for chloride to pass through. Picrotoxin is sometimes used in research. Sometimes it's used in research where you want to kind of maybe induce seizures, right? Because obviously if it's an antagonist of GABA-A, it's going to make it easier to excite the nervous system. So it's often used in research to excite the nervous systems of animals to, say, mimic seizures and things. Obviously the plant produces it not as a benevolent, a benevolent organism interested in scientific research. The plant produces it because it hopes that if you come along and eat its seeds, which it doesn't want you to do, that you're going to get all fucked up, right? You're going to start seizing and convulsing because your nervous system is going to become very hyper-excitable. So it's like many of the compounds that plants produce as herbivory deterrents. So again, I just want to reiterate one last point here, which is, again, just drawing your attention back to the fact that all these different antagonists and agonists are all binding to different sites on that GABA A receptor, right? And so this actually allows for their effects to be kind of additive, right? Because they're not competing all for the same handful. Well, they're not all competing for the same binding site. They're binding to different sites, and so you have some pretty complex kinetics, shall we say, here for this receptor. Let's move on now to the next class of neurotransmitters. I want to talk next about the monoamines. Right, we said last week these are defined as a group because they have a single amine group within their structure. So we'll start with the catecholamines. So dopamine, norepinephrine, epinephrine are the three major catecholamine neurotransmitters. Again, if we just look at their structure, we can see they each have a single amine group within their structure, so hence the amines, hence their monoamines. And then the other part of the catecholamine is catechol, which is this structure here in the green box. It's this aromatic ring with these two adjacent hydroxyl groups. That's catechol. And you can see that that structure is present in all three of them, dopamine, norepinephrine, and epinephrine. Right, so that's what we mean when we say catecholamine. We're talking about how they have this catechol in their structure and a single amine group. All right, what I want to talk about first here is, I guess I'll start with just, we know a lot about these neurotransmitters already. Dopamine we talked a little bit about last week, its role in the brain's reward pathway. We're going to talk a little bit more, I think, probably on Thursday, about its role in diseases like Parkinson's and just in like motor control. Norepinephrine and epinephrine we're very familiar with from much earlier in the course. We know that these are the major neurotransmitters of the sympathetic nervous system. So just to kind of give that context, what I want to talk a little bit about next here is just how do we make these catecholamine neurotransmitters? Like, what is the biosynthetic pathway for their synthesis? You'll notice that they're all derivatives of the amino acid tyrosine. Okay? Tyrosine itself is not a catechol. You can see that it only has that single hydroxyl group on its aromatic ring, so it's not a catechol. But you'll also notice that when you look at the pathway to synthesizing the catecholamines, the very first reaction is this reaction catalyzed by tyrosine hydroxylase, where you add the second hydroxyl group onto that aromatic ring. And so this turns tyrosine into something called dopa, and dopa is a catechol, and therefore also a catecholamine. Okay? So very simple biochemistry here. Tyrosine gets hydroxylated by tyrosine hydroxylase and becomes dopa. And then the next step here involves an enzyme called aromatic amino acid decarboxylase, which does exactly what it says it does. It comes in here and removes that carboxylic acid group. So you can see that group is gone now, and all we have then are the two carbons and the amino group here, the amine group, and now we have dopamine. Right? So two very simple enzymatic reactions to go from tyrosine to dopamine. If we want to go on past that, if we're not a dopaminergic neuron who's trying to make dopamine, but we're one of the adrenergic neurons who's trying to make either norepinephrine or epinephrine, then we have to continue. The next enzyme here is called dopamine beta hydroxylase, and it adds a hydroxyl group to this carbon here, and that gives rise then to norepinephrine. And then the very last step, if we want to go on to make epinephrine, is we have this enzyme here called phenylethanolamine N-methyltransferase, PNMT for short. And it's going to obviously add a methyl group, hence it's a methyltransferase, and there's that methyl group added on right to that terminal amine. So even going from tyrosine to epinephrine, which is the longest of the steps in the pathway here, we're only talking about 4 very straightforward chemical reactions to go from tyrosine to epinephrine. The other group of monoamines are the indolamines. So the classic example there would be serotonin, right? And so again, you can look at the structure of serotonin and you can see this amino group down here, right? This amine group that makes it the amine of indolamine. And then also it has this structure called the indole group within it. And so that's this aromatic ring here that's bound to this little 5-member nitrogen-containing ring. That structure there is indole. So if you have indole and you have an amine, you're an indolamine. So serotonin is the major indolamine neurotransmitter. We talked last week about they stopped the pathway here at serotonin, but you could continue this pathway on to make melatonin, which is also an indolamine. If we look at the biosynthesis here, we can see that serotonin is produced using tryptophan as its starting point. So this is another amino acid, which you can see itself is already an indolamine. It's got both the indole and the amino group. Two very simple biochemical reactions to turn tryptophan into serotonin. The first is catalyzed by tryptophan hydroxylase. So we're adding a hydroxyl group onto the end of the indole here, as you can see. And then we have that same enzyme we saw above, the aromatic amino acid decarboxylase that comes in here and removes that carboxylic acid group. And now you have serotonin, which, as we had also mentioned last week, its abbreviation is 5-HT, which doesn't seem very sensible at the start, but it's because the chemical name for serotonin is 5-hydroxytryptamine, right? That's where that 5-HT is coming from, and last week we talked about the 5-HT receptor that serotonin can bind to as part of that whole presynaptic facilitation process that we learned about. Okay, so let's move on now and just talk a little bit more about what are called catecholaminergic neurons. So neurons that make catechols, so either dopamine or norepinephrine or epinephrine. So the starting point, as we've already established, is tyrosine, gets turned into dopa by tyrosine hydroxylase, gets turned into dopamine by this dopa decarboxylase, so that we already saw on the previous slide. Once you have made dopamine, what happens at that point is we have to load that dopamine into synaptic vesicles. And so again, the picture here is a little bit generic on that front. So I've got this slightly more specific image here. So last week we learned about vesicular glutamate transporters. Well, in cholinergic neurons, you have a vesicular monoamine transporter. Right? So this is a transporter to get these monoamines, like dopamine, into the synaptic vesicle. It works by the same principle that we saw for glutamate, which is that there is this proton pump that is pumping protons into the interior of that synaptic vesicle using energy provided by ATP. And so that creates, again, that proton gradient across that vesicular membrane. And then this is where the vesicular monoamine transporter comes in, because it's going to allow those protons to leave that vesicle, thereby releasing the energy of that proton gradient, and it uses that energy released to power the uptake of the monoamine, right? So here we're talking specifically about dopamine, but this same transporter is used for the uptake of all monoamines, dopamine, epinephrine, serotonin, whatever. All right. Once you have the dopamine inside the vesicle, that's where it gets converted into norepinephrine, if you're planning to go that far. If you're a dopaminergic neuron, you're done at this point. You've made your dopamine, you've packed it into vesicles, have a day over. But if you're going to make norepinephrine, then inside the vesicle, that is where you're going to find the conversion of norepinephrine occurs. Right? So actually they're showing us here that this dopamine beta hydroxylase that hydroxylates the dopamine to create norepinephrine, it's inside the synaptic vesicle. So the dopamine to norepinephrine conversion happens inside the synaptic vesicle. If you're just hoping to make norepinephrine, now you're done. But if you're going to go on to make epinephrine, it's a little bit more complicated, because the enzyme that converts norepinephrine to epinephrine is not inside the vesicle. It's outside in the cytoplasm. So you actually then have to transport the norepinephrine back out of the vesicle, back into the cytoplasm, where that PNMT is there. It'll transfer a methyl group onto the norepinephrine, and then you have to spend energy to pump the damn thing back into the vesicle again, right? It's the same process, that same monoamine transporter, but you had to do that conversion of norepinephrine to epinephrine in the cytoplasm. There are lots of hypotheses about why that would be the case. One simple one is just that the methyl donor is out there in the cytoplasm, because it donates methyl groups to other things too, right? So it'd be kind of like if you said, well, I want a hot dog. Right now I'm hungry. Well, there's no hot dog vendor in the building, right? He's outside somewhere, in theory. There used to be a hot dog vendor on this campus, and I am hungry, so I'm thinking about these things. But you wouldn't be able to get a hot dog here in the building. You'd have to go out, right? Because he's out there. He's out there so that people from all kinds of buildings can come and interact with him. He's not just in here. Same kind of thing. You've got this methyl donor that's out there in the cytoplasm, donating methyl groups kind of broadly for different reactions. So the PNMT is out there where the methyl donor is. You have to get the norepinephrine out of the vesicle to the cytoplasm, methylate it, and then pump it back into the vesicles. And so this is the way we can create dopamine-filled vesicles, norepinephrine-filled vesicles, epinephrine-filled vesicles. And what we'll talk about Thursday when we come back is basically how do we get rid of these different catecholamines when we want to end that signal. So yeah, we'll talk about that on Thursday. I'll see you then. What's up?
I had a couple of questions.
Yeah.
It's mainly around like neurotransmitters and GABA receptors.
Okay.
Is the effect of either the antagonist agonist like balanced out by the affinity or like where it binds? Because I was looking at the picrotoxin and it's binding inside the channel. So I was wondering, does that mean it's more effective?
It would definitely. I mean, I think the idea there is that if you had picrotoxin, and I don't know for sure off the top of my head, but I think you're right that picrotoxin, like, you couldn't override its effect with, say, ethanol or GABA, because ultimately it's binding in there and blocking the actual ion from flowing through the channel. So
would it also be effective based on the time that it binds first? For example, because if, like, let's say GABA is already bound, right? We're assuming that if the channel is already open, then benzodiazepines won't have an effect, or ethanol won't have an effect.
well, so they can still. So yeah, they do tend to work in those additive ways.
Yeah.
So even if you've got GABA bound, and then you have some other agonists come in there, it's almost like they're kind of like grades of channel opening.
Yeah.
So it's kind of like if you imagine a door that's like just slightly ajar, so people can squeeze through, or a door that's like wide open. Yeah. You can affect the permeability and therefore how easily chloride can actually move through. So it is an additive effect.
And then with that, this last question is, with the effect of the barbiturates, would it be, are you less likely to overdose if you already have, like, GABA, or like, is the effect less? Like, if you have GABA bound in benzodiazepines, for example, already bound to the effect of a barbiturate, like, you're less likely to overdose, so you're less likely for it to negatively.
I think it's actually the opposite of the thing. That if you already have, like, if it's already
stacking,
they're stacking on, right? So then, it becomes like a lesser concentration would get you to that permeability point that might lead to significant decrease in nervous system activity.
That makes a lot of sense. And does that mean that barbiturates have a higher affinity to binding to?
I think that's probably part
of
it.
That's
Higher affinity and just like I think a greater capacity to probably cause the gate to open. So a better affinity and a better activity. So again, they're very potent and effective. And like I said, that gap between the therapeutic beneficial concentration and like the fatal concentration is too narrow for safety margins. So,
thank you.
Okay. Yeah.
Hey, how's it going?
We'll go for a walk out of here so we create some space for these guys.